Hello again. This panel that we are going to start, would you like to accompany me, please? Thank you. You can have a seat because I would like to introduce you to the people who is here. So it's good to see you. Thank you. Thank you very much. This panel, possible thanks to the support of VIV Biotech, is now going to address different aspects of lentivirus-based gene therapy from manufacturing to application in humans. Here, to guide us through this territory, please welcome Guruj Linazasoro, CEO of VIV Biotech Spain. And we have also the opportunity to listen to Natalia Elizalde, Business Development Director at VIV Biotech. Tol Trimborg, CEO of CellPoint, the Netherlands, is here with us. And at last, Massimo Dominici, scientific founder of Regeneron, Italy. So the panel is here, our speakers as well. It's yours. Thank you.
Thank you. Thank you, Cristina, for this nice introduction. So it's a big pleasure to be here in Biospain, in this nice city, and with 3 experts in the whole manufacturing process for gene therapies. So everyone has a different field of expertise, so I think we have the opportunity to have a wide view of the major issues and the major challenges in this development process. So, and first of all, let me say that Natalia is going to make a brief presentation of VIV Biotech, then Tol and then Massimo will do the same. So, Natalia, do you want?
Okay, so thank you very much. Well, and thank you very much also to AceBio, to Biospain for inviting us to be here. It's a pleasure also being in Pamplona, where I was born, and well, and to have the opportunity to introduce the company. Okay, the presentation, please. Okay.
No.
Perfect.
Okay.
Yeah. So, well, VIV Biotech is a company fully focused on lentiviral vectors. As you know, lentiviral vectors is a gene transfer technology to insert, modify, or delete genes with the final objectives of curing diseases. We are a GMP CDMO, fully focused on lentiviral vectors from scratch to the manufacture for preclinical, clinical, up to commercial stages. We think it's very important working continuously on the enhancement of vector properties, and that's why we do have an R&D department, as well as downstream and upstream optimization departments working on optimizing the process from the productivity point of view, but also from the purity perspective. Okay. Why do we think it's so important working from scratch? Because we are having into account aspects such as regulatory hurdles, avoiding them, scalability, cost effectiveness, among others. Here you can see marked in green the different countries we are working for, so it's States, Europe, Asia, Israel, and Australia, in compliance with both EMA and FDA. Indeed, the main market is the States, where we do have 70% of the customers, well, they are based there in this country. In total, we are working for 35 international projects. Which kind of projects are we working on? Well, different kind of projects, but approximately half of them, they are for rare, ultra-rare diseases. The other half more focused on CAR-Ts, TCR, so immuno-oncology, and the other half some others for in vivo applications. So in such a case, the lentiviral vector itself is being used as a final product, for instance, for an hepatic disease, ophthalmic disease, etc. Okay. Some of our customers, here you can see, for instance, Cyphos. This is a company working on CAR-Ts in the States. Emendo Biotherapeutics, they are based in the States as well as Israel for an ophthalmic application. Celuna in Norway, these are NK TCRs, very interesting as well. Novavita Therapeutics in vivo application for an ultra-rare disease, in this case for a liver disease. And very importantly, well, we have the opportunity to have here with us today Tol, Massimo, Tol as CEO of CellPoint, a company based in the Netherlands working on CAR-Ts, and well, Tol will introduce it to you in further detail. And then Regenera, okay, that here with us is Massimo, founder and CSO, as well, working in a very interesting approach by transducing mesenchymal stem cells, in this case, for cancer, okay? So, as a company, our headcount has been growing in a substantial manner, and indeed, now we are almost 85 people. We will end the year for sure by being 85 people, and obviously the expertise in virology is key, the scientific background, as well as the experience in GMP manufacture. So, the 4th of June, the opening ceremony of our new building in San Sebastian took place, and this new building is allowing us giving answer to a greater number of batches, as well as larger batches. So here you can see, for instance, the scales that we do have available or that we will have available in the medium short term. So these scales are very adequate to cover the demands all along the development of a given gene therapy. We do have batch sizes that are very good, for instance, for developmental stages. The 10 square meter you can see there is very appropriate for phase 1-2 clinical trials, 30, 60 square meters for advanced clinical trials, and when we are talking about 200, 600, they are more linked to commercial stages. So the process, I will not explain it in greater detail, but this slide just tries to summarize the complexity of the process itself. That takes more than 3 weeks. But importantly, in order to be prepared for a GMP production, you need at least like 6-7 months of preparative activities. And once the GMP batch comes, it will take more than 3 weeks for manufacturing and approximately 3 months for analytical characterization. So you can imagine the complexity behind all these projects and all the expertise that is needed in order to be prepared for them. And finally, well, this short video, okay, just to conclude and to show you a little bit about, well, the new installations in San Sebastian and so on. So, as mentioned, well, fully focused on lentiviral vectors, working along EMA and FDA standards and working globally for different customers in different continents. This is 3,000 square meter facility, which is great because it allows us manufacturing up to 80 batches in reactor per year. But still, we are maintaining this kind of customized services from very early stages, and that's why we do have the R&D department that is giving services to the customers and also doing internal R&D that will be transferred later on to the services. Process optimization, both from the upstream and downstream point of view, so higher, tighter, and also purer vectors. 7 manufacturing suites, able to be answered to preclinical, clinical, up to commercial stages. Quality control, very important to analyze these viral vectors that are being produced. So a great part of the analytics, they are already in-house, warehouse, much bigger than the one we had before that is so important for GMP manufacturing. A complex technical area to give answer to all that is needed to manufacture lentiviral vectors. And then, of course, the administration with the different departments associated to the company itself. Okay, as well as this common area, as we will need more space for these new hired people. Okay. So, well, thank you very much. I hope it was useful as an overview. We'll be pleased to answer any question after this presentation if needed. And of course, if you would like to contact me or at VBiotech, we'll be pleased to continue discussions. Okay, so thank you very much.
Thank you, Natalia, for this presentation. Thank you very much. And now the next presentation is Tol Thummer, who is the CEO of CellPoint, and is going to speak about the company.
Thank you. Will come up automatically?
Yes, it comes.
Well, first of all, thanks very much and to the organizers for inviting us and having us here in this wonderful city. And I'm also really glad that we see some faces in the audience again. So it's a pleasure to be here in Spain, and especially in Pamplona. So CellPoint is a biotech company from the Netherlands focusing on developing CAR T therapeutics. And we are doing that differently compared to others because we want to bring CAR T to the point of care based on a fully automated and closed manufacturing system. I'll come back to that in a second. So we've brought down the manufacturing time to only 6 days, giving us the opportunity to treat patients within 6 to 7 days. And by having this set up this way, we think it becomes plug and play, and I'll explain that also a little bit later. We have 3 programs currently, CD19, BCMA, and CD37 CARs, and we do that in close collaboration with Vive, and we're very happy with that collaboration because it's very successful thus far. Most importantly, the closed and fully automated manufacturing system we do on the basis of a machine made by Lonza called Cocoon, which allows us to bring CAR T therapeutics to the point of care. We're currently roughly 30 people in Leiden, and we're funded by a Swiss venture capital fund called Andy Capital, with a Spanish link actually. So the partner that did the deal was Dani Bach from Spain, Barcelona. So CAR T therapies, I'm assuming most of you know what CAR T therapies are, but it's sort of reprogramming cellular immunity, so the T cells, and this has been very successful over the past year. So there are the two most promising programs are CD19 and BCMA CARs, developed by now big pharmaceutical companies, originally by smaller biotechs, but they were all bought by Gilead or by Bristol Myers. And in the middle you see this lady with whom it all started. Let me see if I can. I don't see it anyway. But this is Emily Whitehead. This was a patient that was treated, I think now 9 or 10 years ago, with only one treatment with CD19 CAR. She was critically ill, almost dying, and now after only one treatment, she's still alive, happily living, and no cancer for almost 10 years now. So from a clinical point of view, CAR Ts are working really well, but a lot of patients cannot be treated yet, and there's a number of reasons for that. First of all, there are limited amount of manufacturing slots. So although there are a lot of patients, and that you can see that actually in the graph below, the red bar shows the total number of patients that are eligible per quarter, and then in black you see what the current companies have been able to treat thus far. So only roughly 10% of the patients can be treated. Also the long vein-to-vein time, it takes the other companies on average 6 to 8, maybe even 12 weeks to take the cells from the patient and give them back to these patients. Some of the patients simply die because they cannot wait that long for this treatment. And the costs are really high, over 300,000 euros per patient. So all these factors limit the current success of CAR T therapeutics. And this is explained to a large extent by the different manufacturing model that the other companies have, called sort of the central model, where you take the T cells from a patient, you freeze them, so cryopreservation, then you have to ship them to central facility sites, either in Europe or in the US. The cells are then manufactured for 2 to 3 weeks, frozen down again, shipped to the other side of the world again, and then given back, thawed, and then given back to the patient. So a long process, a long time, a lot of logistic problems, and therefore very expensive. So we want to change that around by fully automating manufacturing and doing this locally at hospitals. So at the point of care. So basically taking fresh cells, put those cells into a machine. 6 days later, we take out the cells and give them back to the reprogrammed cells and give them back to patients. And we can do this because Lonza has developed a machine called Cocoon, which is on the left side, this futuristic-looking manufacturing module. It's relatively small, and basically we've set up our manufacturing process in this machine, and currently takes 6 days. We do this in collaboration with the top clinical sites in Europe for now. We also want to move this to the US as soon as possible. But we have now initiated this in Barcelona, Antwerp, and Amsterdam. And other machines are being placed now in other centers in Leiden, Belgium, and also Germany and other countries. And another critical important component is to capture all the data and to make sure the quality of our manufacturing process is really high, to be able to present that to the regulators, FDA in the US and the EMA in Europe. So this is an overview. This is still an artist's impression. Cocoons are now tabletop, but Lonza foresees in the future that these cocoons will be on sort of a pole. So depending on the size and the height of a room, you can very efficiently place a number of these machines in hospitals. So you don't need large and expensive clean rooms to be able to stack a few of these machines. We have a very nice relationship with Lonza. So Lonza will, for free, place these machines in the hospitals that we collaborate with, and not only during clinical trials, but also during commercial stage. These machines are regulatory approved and fully up and running. This is just a short slide to show how we do this. We've been able to reduce the manufacturing time to only 6 days. It's a very short and mild manufacturing process. So we believe it's also resulting in a better product. And in this way, we have been able to generate a very short vein-to-vein time. Again, these are the sites we're working with, some of the top sites in Europe, which already are known to work with CAR-Ts, and also have a cell and gene therapy facility. This is a little bit about Excelit, our RT platform. It basically end-to-end captures all the data, ensures the chain of identity and the chain of custody. So it's really important that products cannot be mixed up from patient to patient, and we capture all the data to present to the regulators. So as I said before, we are working on 3 programs. We have our CD19 CAR to treat ALL, NHL, and CLL. And we have filed with the regulatory authorities in 3 countries now, in Belgium, Netherlands, and Spain. We have approval already in Belgium, and I think Netherlands and Spain will be very sure to follow. And then the 2 other programs, BCMA and CD37 CAR, will follow in a short time. And as said, the viruses on our 2 main projects are being produced by Vive. Well, this is the team, and just to show that we have an experienced team that has been active in the cell therapy space for a number of years. And we were able to attract some people from Kite who have brought products to commercial stage before, and that are really key now to help CellPoint to bring our products through the clinical stages into commercialization. So in short, we are going to bring CAR-T therapies to the point of care to make them more accessible to cancer patients that are critically ill and cannot be cured now. We are going to do this by a very short manufacturing process based on Cocoon, so at the point of care. And we feel it becomes plug and play. So the machine will remain in the hospitals, and the only thing that we have to do, the process will remain the same. The people that are operating the machine will remain the same. So everything is the same. The only thing we change is adding a different lentivirus to the system on day 1. And then you either get a CD19 CAR or a BCMA CAR to treat the patients with different cancers. Again, we do this together with Vive, and from the manufacturing point of view, we do this together with Lonza, who actually installs and deploys the cocoons. So again, thanks for having us here, and open to any questions later on.
Okay, thank you. So thank you for this kind presentation and the introduction of your company. And next speaker is Massimo Domenici. He's the founder and CSO of Regeneron, an Italian company. So Massimo, please.
Good morning. Good morning, everybody, and thanks for the invitation. It's, I would say, it's a nice feeling to see people around after 2 years of non-meeting in person. So I'm a physician. I'm between academia and companies, and I founded this Regeneron company 2009, and we are based in Modena, in Italy, which is very famous for car engineering, I would say, and manufacturing for balsamic vinegar or Lambrusco, not much for gene therapy. So we are trying to change that flavor, and we do it thanks to the collaboration with the University of Modena. So the company has a vision, which is I think is shared with the what has been said so far by previous speaker. So we wanted to change the natural history of some diseases, some still incurable cancer. And so we do it with the mesenchymal stromal cells, and either from a therapeutical point of view and also from a diagnostic point of view. Also, we have a CAR-T project that I will be briefly mentioning during this quick call. The company has 3 main pillars, essentially a product for solid tumor, a gene therapy product. The company has also developed the possibility to work with others to manufacture cells, a CDMO, I would say, approach. And then the third pillar is to provide a medical device that will allow the diagnostics, mostly in oncology, but not only in oncology. So this is the company. We have about 500 square meters of classified environment where we can manufacture cells for ourselves and then for others. We have a candidate for cell and gene therapy, which is RR01, which is unique examples of using mesenchymal stromal cells delivering anti-cancer agent, and particularly we're focused on an agent called TRAIL that we gene modify to make it more bioavailable for solid tumor treatment. And it's relevant for us because TRAIL is very active against very many cancer types, and we do believe that the delivery of this agent with the cells that is gene modified will be a game changer for aggressive solid tumor where the stroma and the environment is relevant for the tumor progression. And this is our pipeline. It's a small company, but we are very ambitious. So we wanted to treat still deadly cancer like pancreatic cancer. This is we are at the beginning of clinical phase of a phase 1/2 study, first in men, first in class, and we are also working on other still aggressive cancer type like sarcoma or mesothelioma, and also we have some other non-oncological targets. And the manufacturing process, it's extremely challenging. It is an autologous product. We are simplifying this process and minimizing the expansion time of about 3 weeks or more or less. And again, here the help of Vive was crucial in this manufacturing step, and a relevant partner since the beginning from the concept to the clinical implementation. We're using lentiviral vector, so we can gene modify the cells very, very efficiently and very safely, and this is also important. We took advantage of our manufacturing facility to generate adipose perivascular cells for the treatment of COVID patient. So we have been approved for a phase 1/2 study, and we already enrolled 3 patients in the study, and with some interesting data. And it's a multinational, it's a multicentric study in which the company is manufacturing cells for the hospital. So we are very happy that we can use an allogeneic-based product, which is also was for us the first time to do. Also, we generated the CAR-T for neuroblastoma, for gliomas, which is an anti-GD2 that we have been developing with University of Modena, and we are somehow transferring into the company in collaboration with other institutes. Of course, we wanted to take advantage of our development, and essentially we are proposing the company as a CDMO for TMPs. This is the layout. We have about 450 square meters of manufacturing step with 5 suites, 2 for gene modification, so biosafety level 3, and 3 class B for cell manipulation. We have R&D labs and the warehouse, and then nitrogen storage for large storage of allogenic product. We have been approved by HIFA, our regulatory agency, since the beginning of pandemic, and now we are certainly happy to listen to possible customers that would like to expand their manufacturing capability. And ultimately, which it's a slightly different topic, but for us was important, and this is why we are proposing also for the market. We have been developing bioreactors that allow the expansion of cells, and we use this for drug testing and diagnostic, also lately for exosome production. Essentially for us was extremely important because we did most of the R&D phase to validate this technology in 3D, minimizing the use of animal models, and then going to the animal models in a later phase. And for this reason, the product is on the market. Also, we publish data on this technology in cancer, and this is a paper that will be appearing soon on Frontiers. And a group in Bologna used this to generate neurospheres for drug testing in the neurological field. And this is how the spheres is growing into the bioreactors. And then on the right, you see how the cells are colonizing the matrix very rapidly. So I'm handing here, I'm happy to be here. I wish I could be here in July to see the most attractive part of Pamplona. Thanks Invive for the invitation, organizers, and happy to take any question during the next minutes. Thanks again.
Thank you, Massimo, for your brief introduction to Regeneron and to be on time. I think we have 20 minutes more or less for some questions. I don't know if anyone in the audience wants to ask any question. Feel free or tell me, or I don't know. But I have prepared some questions. The first one is quite open, and I will appreciate very focused answer according to your field of expertise in this production chain. And in your opinion, your view, which are the main aspects to be considered for the production of a gene therapy from your area of expertise? Please, I don't know, Natalia, we start. Tor?
If you want, I can start. If you want, I will focus on the lentiviral vector field in this case. So from our point of view as a company, and as mentioned before, what is key is at the beginning, focusing also on regulatory aspects. Well, as an example for lentiviral vectors could be the fact that different antibiotic resistances could not be accepted later on, or different tags that you should avoid for GMP. So having into account that from the very early beginning can avoid a lot of different hurdles at the end of the day. So this is key. Also, scalability, because if you are developing a given process that is not scalable, at the end of the day, it's not worth working on it, because you will end your phase 1, but it will not have a future. And I guess you share, because your philosophy is very similar, but also cost effectiveness. So it should be cost effective, and also the more patients are reached, the better. So this is how we think. Yeah.
Tor?
Yeah, so I agree with Natalia. So also from our point of view, the regulators and taking them along, and so lentivirus is one of the key parts in our manufacturing process. There is, and they're building on that experience. There are a number of companies that have products approved, so the bar is quite high. So that's why we're again very happy to work with Vive to work closely with the regulators, and our approach has been to involve them very early on. So we've already had meetings with Dutch and Belgian regulators. We are in the US, asking for a pre-IND meeting, and to really take them along in our way of thinking and how we want to bring CAR-Ts at the point of care. So that's, we have this additional layer in addition to Lenti, but also the additional layer of bringing manufacturing to the point of care with hospital or gene therapy facilities at hospitals becoming our manufacturing sites. So we have to educate them, and we are actually not surprised, but talking to the FDA with my previous company, but also now with the European regulators, they're very open and they're very helpful and constructive in working with us to try to overcome some of the problems. So I fully agree. Cost effectiveness is key. Bringing in the most efficient way these cells, or reprogramming these cells in the most efficient way and cost-effective way, will be critically important to bring our view of CAR-Ts at the point of care and make that a success. I think for us a key will be also getting all the various academic centers from a quality point of view up to speed and up to par. But that's why we try to make our process as simple as possible and really prove to do this in multiple centers but still at a very high quality.
Massimo?
Yes, well, I think I can speak from either, I would say, a developer, academical developer, and a corporate consolidator, I would say. And the suggestion, I mean, in my experience as an academical developer, the major point to consider is that you need to value your research in a GMPification scale-up. I mean, consider that all the discoveries that you are doing in the lab, in the academia, have values if you're able to GMPify them under a sort of quality control point of view. It's not easy. It's not part of the DNA of academia, but it has to be part of the DNA of academia. Otherwise, you might develop incredibly good product in big journal, but at the end of the day, you are not able to impact that on a clinical point of view, or the attrition rate for this impact to be so long that will be very poorly feasible. So that is, I would say, a consideration as an academical developer. As a corporate developer, I would say the suggestion to expedite the development is to partner with CDMO, if you need a CDMO, or with the key reagent provider like Viver in our case, very early since the beginning of your development, because that is where you seed the plant in the right way. If you're seeding the plant in the wrong way, then the growth of the plant will be a problem. So try to seed as early as possible with the CDMO, with your vector providers, because in this way you're going to fix the problems since the beginning, and then you're going to smooth going to the clinical phase. Otherwise, you might find a problem. If you are doing this later on, then you might have the plant that has been growing a little bit more than you want, but maybe is not growing in the right direction.
Right, that's fine. I think that both of you have mentioned cost effectiveness, and I think this is related with a major challenge of gene therapies, which is the global accessibility. I think, in fact, Nature have a recent editor's note about this issue. And do you believe that the key question is to improve cost effectiveness, or there are any other things we can do to guarantee this global accessibility for gene therapies to any patients around the world?
Yeah, so I hope to think that our way of bringing cars to the point of care, and we start with cars, but we also with T-cell cars, but we also want to develop into NK and towards solid tumors a little bit. Of course, we hope that our approach by bringing this at the clinical centers and also involving the clinicians and maybe also coming up with a different business model together with the regulators, which will allow us to do a little bit simpler and easier, more studies. It's not easy, but in the US there, for example, is the opportunity to file for a multiple IND where you actually do one registration of multiple products. So that could maybe speed up ways of doing smaller studies quickly. I think we really hope that our model brings it a bit closer to the hospital and also, well, again, in a cost-effective way, but also in a more scalable way. And in that way, try to resolve part of the issue. But there will be multiple. Allogeneic will definitely also have its own space. So there will be, and there will be new developments.
Yeah. From my point of view, cost effectiveness is a key issue for sure. And there are different ways of reaching it. I mean, one is scaling up, having bigger reactors, and therefore the costs for sure will be lower. But there is a second part that is even more interesting from our point of view as a company that is investing on R&D to improve continuously the processes, so making them more and more cost effective. That will have a direct impact on the final therapy cost and the accessibility to the patient. So that's why, for instance, the R&D team is working that much on trying to enhance the titer by modifying the cell line, so more production is reached by having the same reactor size, or even pseudotyping strategies to really target the cell and therefore decreasing the amount of lentiviral vector that is being needed for a given therapeutic effect, among others, or your initiatives as well. So our common objective as an industry should be reach more and more patients, and the only way is going through this way from our point of view, yeah.
Massimo, any comments? Or?
Well, I think the major word that I would use to somehow get into a cheaper product is the word confidence. The confidence of the developers, the confidence of regulators, and the confidence of the market that is believing that these products are a game changer. The confidence has to be reached in the time. It's part of the evolution. I think we are at the same time lucky and unlucky, because lucky in the sense that we are living this moment maybe without being aware of living this moment, but we are essentially living a revolution. And all the revolutions have cost. In many ways, the cost that we are spending here is the time, as you outlined, and the cost, I'm talking about economical cost. So if the confidence will be increasing, then, for instance, the cost of all the quality tests that we are doing in the product will be decreasing. And these are major part of our cost. So the confidence is related to the decrease of this type of test and cost of test. And the confidence also of the public healthcare system that in Europe are paying for this treatment has to be increased, so that there will be somehow more, there will be more flexibility in the reimbursement, and then this will be certainly decreasing the manufacturing cost and ultimately arriving to a large amount of patients.
Right, okay. Well, and I want to change a little bit the market. The last data of the market show that the market is growing. It's growing and growing and growing. And probably this will influence at the end of the day in the prices and in anything, no? What is your opinion about this tendency of the market? Is going to follow growing and growing and growing, or how do you see the situation?
In this case? Yeah, the market is growing, this is clear. I mean, there are data there that reinforces this fact. For instance, We know that last year, 2020, it was like $20 billion raised just on this advanced therapies field, which is a lot. And even if we compare it with 2019, it's like double. So the market is growing. This is clear. The trend tends to be this way as well. And also it's true that the market is being defined now. And what we are seeing or what we are observing is that, for instance, more and more allogenic approaches are being developed. Okay, autologous, of course, they will continue, but allogenic approaches are there. In vivo applications, in this case of lentiviral vectors, even in vivo applications with these pseudotyping strategies to target the final cell, so decreasing the amount, but at the same time being very efficient. This could be the second trend, and then solid tumors. So far, for instance, you were mentioning solid tumors approach, but so far it's true that CAR Ts, they were always thought to be used for hematologic cancers, and now more and more solid tumors approaches are there. So I think that it will continue the trend towards an increase in this market, at least in the coming years. Yeah.
Tor, do you share this?
Yeah, definitely. I mean, most of the time, from my experience, these market reports are pretty okay, and it is good if the market is growing. And I hope that it grows because we will be able to treat more patients at a lower cost. So, but still increase the market. But I fully concur with Natalia. So, cell therapy has. I also have an investor hat. So one day per week I work for an investment firm. So, and I did that already earlier when I just left university. And then cell therapy, until probably 2013, 14, 15, especially in Europe, was very difficult and also difficult to get funded. But it's here to stay. As I showed in my presentation, the big pharmaceutical companies have jumped on it. So the 19 and BCMA CAR are owned by 4 of the largest pharmaceutical companies. All the others have invested in it and are heavily investing in it. And it's just at the beginning, where antibodies were 30 or 40 years ago, early 80s. That's where we are now in cell therapy. So the success of 19 and BCMA has been extremely important for the cell therapy space in general. And then there will be a lot of new developments, getting CRISPR-Cas and all these new techniques up to running, and maybe eventually get. I'm not a big fan of allogeneic, so I think especially the way we do it, bringing the manufacturing time down and bringing the cost down, we solve the same problem in a much better way from a product point of view. So, but there will be space for allogeneic. We will move slowly from hematological indications to the more difficult solid indications. But I think Massimo has a very nice approach already towards with GD2 towards some of the brain tumors. So there are early developments there, and those, of course, the much bigger market opportunities. And if they're going to mature, which they will, most likely in combination treatments, then the markets will definitely. I think it will even grow bigger than maybe foreseen.
Massimo, your view?
Well, I concur that the market has a positive outlook and is growing, which is I think is excellent. My point here is the market of the manufacturing is growing. The patients are out there, so I think from let's say usability point of view, I think we need to be sure that the hospital will be ready for this revolution, because that is something that we should be considering. Physicians, nurses. So next to the market growth, which is great, I think we should consider also the training of the healthcare providers, which has been somehow neglected. And the training of people that will be taking part of this revolution from a manufacturing point of view. It's not easy to find people, good people that are actually putting hands on cell therapeutics. So I would say that marketing is growing, but these numbers are suggesting that we need to be ready for that, either in preparing the next generation of manufacturer and the next generation of physicians that will have, and nurses that will have to take care of this treatment, their complication, their follow-up. If we don't do this, then I think this imbalance would be a funnel into a real implementation on cell therapy. Of course, I'm solid tumor biased, but I see this as an issue.
Okay, thanks. I think we have to finish because we are on time right now. So thank you very much for your nice presentation, for your contribution to this dialogue. I hope that the audience has learned a lot about the production of gene therapies and about also the major challenges we have to face in the next future. Thank you very much. It's a big pleasure. Thank you.
Thank you.
Thanks. Good.
Thank you. Thank you very much for shedding light on gene therapies development and what they can do for us. Thank you very much. Now it's time for a 30-minute break. We'll be back at 12:15 when we'll be addressing such relevant topics as precision medicine in modern psychiatry and neurodevelopmental disorders and the challenges we face tackling cancer. So thank you and see you at 12:15. See you.